moving from the anatomy of the microscopic into a little more of a physiological aspect of how it all works. We need to start with the sarcolemma. Now, the sarcolemma is the membrane around a muscle cell. So at rest, so before contraction, when a muscle is just in a relaxed state, that plasma membrane, that sarcolemma, is said to be polarized. Now, the term polarized means there's a difference of charge across the membrane. In this case, the inside is going to be negative relative to the outside. So again, this is at rest. So the cell's not doing anything, it's just at rest. The inside is going to be negative relative to the outside. So what that means is, if you had a voltmeter, and you took that voltmeter and put the probe through the membrane, you'll notice that the inside would have a negative charge in comparison to the outside charges. Now, I keep saying relative. It's relative that the inside for a skeletal muscle cell is negative 70 millivolts lower than the outside. Now, to be clear, that doesn't mean it's an absolute number. It's not like the inside is negative 70 and the outside is 0. This just means the inside is 70 millivolts lower than the outside membrane charge. So it theoretically could be outside could be 100 millivolts. Inside is 30 millivolts, 70 millivolts lower. It could be the outside is 0. The inside is negative 70 lower. So this is a relative number. It's not an absolute. It just means the inside is 70 millivolts lower than what the outside charge at that point is. And this is all at rest. So when that muscle cell is at rest. To start everything off though, to have the initial signal, you have to start with a motor neuron. Now, a motor neuron is part of the nervous system. It's what's going to carry the action potential or carry the signal from the brain and spinal cord out into the muscles. So it's taking that impulse and bring it down to the actual muscles itself. Now we'll have these synaptic vesicles in the end of this motor neuron. A synaptic vesicle is going to be a vesicle, just a held container. And in this case, we're going to have acetylcholine contained within. Now, there are many types of neurotransmitters. We're going to focus on acetylcholine because acetylcholine is what is used when the neuron reaches the muscle, called the neuromuscular junction. So we're going to focus on this one particular one, but there are many types out there. So each branch of one of these neurons, so this motor neuron comes down, can have all these different branches. Each individual branch that touches is going to form what's called a neuromuscular junction. That's the junction where you have that end of the neuron coming to the actual single muscle fiber. And this can happen for many, many fibers and many, many neurons. But this is where the signals are transferred from the nervous system to the muscular system. And it's transferring through the end of the nervous system, end of this neuron, called an axon bulb, or an axon terminal. It's the very end part, right where the neuron stops before the muscle starts. Now, the muscle that's going to be just right around that bulb, is called the motor end plate. So the motor end plate is a specialized portion of the sarcolemma. It's going to be wavier to help increase surface area. More surface area means more receptors. So we're going to have this axon terminal from the neuron coming down into the motor actual part here, right into this plate. So right in the motor end plate. And what's in between, because they don't touch, there's a thin area in between. That's known as the synaptic cleft. Now this synaptic cleft is fluid filled. So that means the axon terminal from the nervous system comes down doesn't quite touch the motor end plate, but there's fluid in between it. So here's what it looks like. This is the actual motor neuron coming down. You can notice in this particular drawing, they have 3 different actual axon terminals. Here's 1, here's 2, here's 3. Now, there can be more, there can be less, depends on the situation. But each one of those is going down to the motor end plate of the muscle. So what we have, first of all, you must have a nerve impulse. You must have what's called an action potential. That's going to travel down that motor neuron to reach the axon terminal, the end port. Now, that action potential can't just go across, because an action potential is pretty much the movement of ions, but this difference of charge. We'll get more into what that is in a second, or a few minutes actually. But think of it as a very small electrical charge here. What happens when electricity hits water? It just dissipates. There's no easy way to control it of where it's going. So we have to take this action potential, this electrical charge, and convert it into a chemical signal. The reason you want to convert it to a chemical signal is you know that chemical signal has a little more control to it because it is governed by the laws of diffusion. It'll go from high to low concentrations. You kind of can predict where it's heading. So we have to make sure that this signal coming out of the somatic nervous system, think voluntary nervous system, but out of the somatic nervous system, comes all the way down to the end to that axon terminal, that action potential is being carried. So it reaches the end. It's referred to as the neuromuscular junction, where the neuron and muscle meet. First thing that happens, that action potential is going to trigger the voltage regulated calcium channels. Basically what that means is these channels are closed unless an action potential reaches them, then these channels open up. So they're triggered by the action potential, the depolarization caused by the action potential. So once they open, in this case, calcium is being released. So we're going to release calcium into the axon terminal. Now, that calcium, once it enters the axon, is going to cause the synaptic vesicles to fuse down at the end. So it's causing them, instead of just floating around, they're now all going to the end to fuse, getting ready to release their contents. Once they fuse, acetylcholine, ACH, is acetylcholine. So once those vesicles fuse, they open up and release via exocytosis, so releasing their contents out into the synaptic cleft. Now, as they release out into the synaptic cleft, skipped one, the acetylcholine is going to diffuse across synaptic cleft, because right in that vesicle is an area of high concentration of acetylcholine. The synaptic cleft is a lower concentration area. High diffuses to low. That means the acetylcholine will diffuse out and across, going to the motor end plates. Once the acetylcholine reaches the motor end plates, the acetylcholine molecules will bind to the chemically gated channels, or ligand gated channels there. These channels are closed unless the proper key is there. In this case, the proper key is this chemical acetylcholine. Once acetylcholine binds, the channel opens up, allowing ions to pass through. Sodium and potassium will diffuse through this ligand or chemically gated channel. What happens here is sodium diffuses into the muscle from a gradient of high outside concentration to low concentration inside. So it fuses into the muscle, while potassium is diffusing out of the muscle cell. Potassium was in a higher concentration inside, is now diffusing to a lower concentration outside. but they don't move in the same ratio. It's not a one-to-one exchange. Sodium is moving faster. That means because sodium is moving faster, the inside that was at rest, negative 70 millivolts, you're adding more positive ions in than you're taking out. Let's say a hypothetical, 3 sodium in for 2 potassium out. That means the inside gained 1 positive charge. And as they keep flowing over and over, that negative 70 inside becomes negative 69, negative 68, negative 67. It's depolarizing. The inside is becoming less negative. Now, this is only a small event because there's not too much of a difference here. So this is a very slow depolarization in relation to other depolarizations. So initially, this local event is called a graded potential because it just happens right around the actual receptor. But if enough acetylcholine was bound and that channel state was open long enough, and other channels around it are also opened, they might all kind of add together to create a larger change, a larger depolarization. If enough happens, then, and you reach this threshold, so there's a certain level where if you reach this, you immediately can create an action potential. So if enough of this change slowly at negative 69, negative 68, negative 67, 66, 65, if you keep going up and keep adding together to hit threshold, then you can immediately create an action potential. That action potential is the same signal that was originally in the motor neuron. You've come full circle. You start with the electrical signal, which is the action potential in the motor neuron, convert it to a chemical signal, acetylcholine in this case, convert it back to an electrical signal, a graded potential, and then if it all adds up and hits threshold, back to an action potential. Now this action potential will spread in all directions as long as it can. Think about it through a pebble in a pond. That ripple goes in all directions. That is the action potential. As long as the areas around it are negative comparison to the outside positive, so remember, they're at rest. As long as the areas around it are at rest, it'll go in a full 360. If any areas aren't at rest, they already have reached the threshold, it can't go that way. So it can only go into areas that were at rest. So now let's take a look at it in picture format. We've gone through all the words, gone through the descriptions. Let's look at what visually looks like. We have the action potential coming down. It's traveling down the motor neuron. It's going to reach right here, the neuromuscular junction. Now that is blown up over there. So in the blown up aspect now, that is the neuromuscular junction, the signal comes down. As that action potential comes down, it's going to trigger the calcium-gated channels. Calcium will rush into. So now calcium's flowing into the actual neuron. That calcium is going to cause this synaptic vesicle to fuse with the outside membrane. When that fuses, you can see all the acetylcholine is going across diffusion. It's diffusing away from the higher concentration to a lower concentration. As it does that, some bind to the chemically gated channels here. These are chemically or ligand gated channels. If they bind, the channel opens up. Sodium is rushing into the cell. But also, potassium is going to be rushing out of the cell. So you have potassium rushing out, sodium rushing in. Sodium is moving faster than potassium. So you have both moving there together. If enough of that changes, enough of those slight depolarizations, those graded potentials occur, if they hit threshold, you can trigger your action potential and it takes off. So looking at how the physiology of the neuromuscular junction is working. So think about this from a little mini case study second. Botulism. You don't want to get botulism, first off. This condition results from eating foods contaminated with bacterial toxin, the botulism toxin. Now, the botulinum toxin is going to prevent the release of acetylcholine. All right, so the toxin prevents release of acetylcholine. What symptoms would you expect here? Just think about that for a second. What symptoms would you expect if the botulinum toxin is blocking acetylcholine? And then at the same time, we know that Botox is derived from the botulinum toxin. So Botox prevents wrinkles. How do those correlate? How does the botulinum toxin equate to removing or preventing wrinkles? So think about that one for a second. See if you have an answer, an idea of why, if you can block acetylcholine release, you're looking at then what's the symptom of botulinum toxin, but also how does Botox work? Well, if you're thinking about, okay, if we block acetylcholine release, that means you can't release that across the cleft, you can't bind to the actual motor end plate. If that chemically or ligand-gated channel doesn't open, you can't cause any depolarization on the membrane. There's no contraction. Botulinum toxin will prevent contraction on the muscle, preventing the neuron from connecting to the muscle. But how does that relate to Botox? Well, Botox is a lesser amount of it, obviously. You don't want to cause too much of a problem. But it's causing a little bit of the same thing. The muscles cannot have their signals received from the neurons. That means the muscles can't contract, the muscles can't wrinkle. Because a contraction shortens the muscle, that's what causes wrinkles, the shortening of the muscle. But if the muscle can't shorten because it's paralyzed, you can't have the wrinkles. Now, botulinum toxin, can be very problematic if high enough doses are ingested. Because think about it this way. If the diaphragm, right here, the diaphragm stops working, it's a muscle, you're in real trouble really quickly. So botulism a lot of times starts more on extremities, think fingers, toes, face, and then works its way more into the core of the body. Just a little idea of how some applications coming out of this. But now, once that contraction is done, so once you want to stop the contraction, because I don't know about you, but I want to hold my muscle in a contracted phase forever. So you need something called acetylcholine esterase. Acetylcholine esterase, or ACHE, is an enzyme. This enzyme will break down acetylcholine into its two base components. If it breaks down acetylcholine into the base components, it can't work anymore. It can't stimulate the actual receptor to open up and allow the signal to pass through. So it prevents the continued contraction of your muscles. So if you're looking at acetylcholine esterase, this part right down here, you would basically stop those individual ions from being bound. If acetylcholine can't be bound, you can't have sodium or potassium traveling through. That means there's no more muscle contraction. So the best way to stop a muscle contraction is for the enzyme that you already contain just simply functioning to remove the acetylcholine and then recycle, of course. Your body wants to recycle everything. So that's the neuromuscular junction. Neuromuscular junction is going to be taking the signal from the nervous system, converting it from an action potential into a chemical signal, into the muscle, and being converted back to an action potential.